Well, don't be afraid of one microbe being out of whack because it's such a massive community with thousands and thousands of different microbes, and they have so much overlap in what they all do that it's okay if one or two are wrong as long as their friends are still there doing what they're meant to be doing and kind of making up for that slack. So that's kind of the approach that we've taken both in our research and in our industry work.
Somewhere between a baby's first breath and an astronaut's return from orbit, a hidden thread weaves through us all. It's older than DNA and more responsive than genes. It whispers in gut feelings, blooms in diversity, and declines when we neglect connection. Today's guest, Dr. Heather Armstrong, doesn't just study the microbiome, she listens to it. In this episode, we follow that thread through birth, healing, and high performance to ask a question as old as Genesis and as modern as genomics. What are we becoming, and what's feeding us along the way? Dr. Heather Armstrong is a leading microbiome researcher and translational scientist whose work bridges pediatric, gastroenterology, immunology, and systems biology. As an associate professor and Canadian research chair at the University of Alberta, Calgary, all right, fellow Canadian, at least by location, and founder of the Translational Medicine Core. Her mission is to make the microbiome actionable. Heather's research has impacted infants, athletes, and patients navigating chronic disease. Her work is defined by one unifying principle, and that is decoding the dialogue between microbes, diet, and host to preempt disease and empower health. So in this episode, we are going to chat about, well, the infancy of the microbiome science itself, the promise of personalized nutrition, and the beauty of science in the mess of real life. And so welcome, Dr. Heather Armstrong. It is such a pleasure to have you.
It is a pleasure to be here. Thank you for having me.
Well, and so for context, we were introduced via Hannah Dose of AU Genetics. So we got to give her a shout out for making this introduction happen. And, you've got clearly a number of passions underway, both academia and entrepreneurial. And so, I'd love to understand, to the extent that you'd like to share, how does one come into the microbiome? And it's a bit of a loaded question because we're in some sense cut from the same cloth, but I would love to hear your story.
Yeah, for sure. It's funny. I'll have colleagues that, they found their passion in early life were during their undergraduate studies, and they've stayed in the same field throughout all of their studies since. And my pathway doesn't look anything like that. It's all over the place. I started off in physiology and then neurosciences in my undergraduate and doing quite a bit more psychology and physiology. My master's degree was in cell biology and genetics and really trying to understand the basic biochemistry of the human body right from the baby cells. And then my PhD shifted to oncology, but it was really, if you look at the nitty gritty of it all, it was really systems biology. And so trying to understand that physiology, that connectivity of everything and how all these biochemical pathways work together in different cells to shape our health and well-being. And when I finished up my PhD, I really wanted to dive further into gastroenterology. I have inflammatory bowel disease myself. So I think it's a very personally driven passion. It pushed me into the world of gastroenterology and shifting from human cells, human health, and looking at kind of the physiology of organ systems and moving instead into understanding then how we as a human being and all of the cells that make us up interact with these gut microbes and different microbes that live in our body and how that symbiotic relationship can promote health or disease.
Yeah, it's not something that is, or at least it certainly wasn't taught in, let's say, the general science textbooks when we were in our undergrad, right? Insofar as, well, it's a microbial world. And I think, we both know Joe Handelsman, we both Rock Knight.
Lay. These are all pioneers of the industry, bringing to bear the tools of things like metagenomics, and really complex bioinformatics to, as we talked about, reveal the microbiome science in its infancy. But I remember the first time I heard, you have more microbes in you and on you than you do human cells. And, now it almost sounds like a platitude or cliche, but no, it's still pretty compelling. So much so I remember telling my grandmother about it, and it just absolutely terrified her. So I guess it's not PG 13 or 80 as it turns out, but just this whole idea, even to kind of go to the edges philosophically, some have been heard to say things like, well, maybe we're just walking incubators for these microbiomes, or what was a quote I heard recently from one of my guests is we are I think he said cytoskeleton, but I suppose at a macro level, our muscular skeletal system. And you might say, okay, well, that's a stretch, but well, yes and no, because we know there are things like, the gut-brain axis as an example. So literally science research has shown, different profiles of gut microbiomes are linked to the way we think, that is like mental health. So would love to understand what you're seeing, at the tip of the spear there in terms of, gut brain access and, well, really where your passions have taken you thus far.
Yeah, for sure. I think you're completely right, but it's amazing how much this back and forth between the human and the microbes. They can't live without us. We can't live without them. I think a lot of people recognize that this microbiome in the greatest amount is found in the colon, in the large bowel of the intestine, but it spans to so many other regions of the body. And what's really neat even about the gut microbiome, and I think we all recognize this, is, these microbes, they produce metabolites, and those interact with the cells of our gut, and they're taken up into the bloodstream, and they traffic throughout the rest of the body. They interact with so many different organ systems. But in turn, we're really engaged with those microbes too. So the metabolites that are produced by the cells of the gut, the human cells of the gut, the different bile acids and products that are produced by the liver that spew into the gastrointestinal system, how the gallbladder and the pancreas produce all of these different products that end up in the gastrointestinal system. This really shapes the microenvironment of the gut, all the different metabolites, nutrients, the pH of the gut. And so this kind of sets up the world that these microbes live in. So it really is kind of a two-way street. We create this environment that allows them to survive and thrive, and in turn, they're meant to benefit us. But if any of these organ systems end up unhealthy in a human setting, you can naturally see then how the environment that these microbes live in can become quite different, and that can change the shape of not only which microbes are living in the gut, how happy they are, how much they're thriving, and what their functions look like. So then how much are they actually benefiting us? So I think that's really been a key driver of our interest as a group, is understanding what is it about the human body that impacts the composition and functions of this microbial community that lives inside us. And in turn, then how are we impacting what they do for us, how much they benefit us? And so what is it about our environment and our lifestyle? Nutrition's a big aspect of that understanding. We're not just feeding us, we're feeding our gut microbes. So how do we kind of promote the whole system to stay healthy? But other aspects of life too. So how well are you sleeping at night? Your mental health and stress levels, your physical activity, how much is that kind of impacting both the human but also this microbial community? And so how do we maintain this complete health of the full system to not just promote health and prevent disease, but promote, athletic prowess, intelligence, all these other aspects of the human body that I think people are generally searching for. How do we excel and thrive as a human being throughout our lives?
I'm called back to one of the first things you said about your journey in academia and studying all these subdisciplines and, all of a sudden not realizing it, we're like, oh, I'm doing systems biology. And I'm starting to see that, sometimes the way we see the problem is the problem insofar as remembering that, how we look at the body as whether, a specialist or a subspecialist without the context of the greater, system itself, or holobiont, I've heard it referred to. You lose the forest through the trees. And, it is, again, but just kind of push on the systems component there a little bit. And, hey, let's start at the beginning or even before the beginning, if you will, insofar as infancy is concerned. One of the things that just absolutely blew my mind was this notion of the enteromammary pathway. So basically, from gut to breast milk, and this idea of immune sampling in the gut where the mother's, gut mucosa is, constantly exposed to microbes, food antigens, environmental signals, et cetera. And you've got these like specialized immune cells. You've got these, I think, especially, the dendritic cells and B lymphocytes. And essentially these in the Peyer's patches and the mesenteric lymph nodes are sampling, these antigens. And, I'm just on the surface here. I know enough to be dangerous, but the fact is actually living the experience now with my daughter getting the benefit of, the breast milk and from her mother, from my wife. And it's not just that it's breast milk, but it's so, specific to the environment, right? So given this like kind of, as I say, this enteromammary pathway, you've got this incredibly potent, localized, the adaptive immune system from birth. So what's your take on that in your research? And maybe that's a good springboard into, say, how the infant gut is colonized over time. C-section babies, what do you do in that kind of situation? Because, as we know, the first exposure to the birth canal is, your first, in theory, exposure to the microbial world. Maybe that's changed. But, there's the ideal scenario, vaginal birth, breast milk, et cetera. Well, what happens when you don't get that? And what's the substitute if you can't have that? And, I asked this because my wife's a C-section baby, and a lot of folks are, and a lot of moms try to breast milk and they do so, unsuccessfly or with challenge. And so it's like, what do you do? I'm hearing that there's all this kind of richness of, health span and immune system health that, I didn't get, or my baby didn't get it. What am I going to do? What are you seeing at the frontier?
Yeah, for sure. It is such a fascinating field, and it's really just exploding right now, the number of studies being done and how much we're starting to understand. I mean, it's really incredible, that connection between not only mum and infant during the entire pregnancy process, but there are so many of these studies that are even showing, mum and dad's health during that year in advance of even conceiving can have such a significant impact on the development of the infant, that development of the immune system, that exposure of the infant to these different antigens during that pregnancy process and far beyond that. And so it's just incredible to see, how far back you can go before even conception, that these impacts are rampant on the infant. But I mean, if we really think about this whole microbiome relationship and recognizing, I think the number one thing to note first is really this microbiome that we have as a human being, as an individual. We each have this kind of fingerprint version of a microbiome that is our microbiome, and that's really established in the first roughly 3 years of life. And beyond those first couple of years of infancy, we now have what will essentially, with a little subtle changes along the way, be our microbiome for the rest of our lives. And so it's really those first couple of years of life that are the most essential in developing the most healthy version of the microbiome that we can acquire. There are certainly lifestyle changes we can make along the way to promote improvement, but it's really that early life exposure to gain as much diversity in the microbiome as humanly possible in those early years. Now, you've kind of touched on this. There are different things that impact the development of the microbiome in early life. Nutrition and breastfeeding is certainly one, and be it a vaginal birth versus a cesarean birth, that's another. Any environmental exposures that you receive along the way. Are you exposed to antibiotics? Are you playing in the dirt? There's so much opportunity to develop this microbiome in early life. And I think one of the big take-homes that society needs to kind of get a hold of again is that there's no perfect path, Things are going to happen, and it's really quite a miracle that any of us are born in the first place with all the incredible things that can go wrong. And so, as parents, not shaming ourselves if it hasn't gone perfectly to plan, and just recognizing that, it's incredible that we have all these modern medicine opportunities for a baby to still be born, for mum to still be safe. And, any early life infections, we can actually cure those and allow the baby to continue to survive and thrive. These are all incredible opportunities, and to not be kind of shameful or stressed or concerned about the fact that things aren't going to plan. So I think that's really a big number one take home. Now, what do we do to kind of recognize that these exposures will shape the microbiome and development of the immune system and cognition and the nervous system and everything else throughout those early years? There are quite interesting studies that look at cesarean birth versus a natural vaginal birth. And there are certainly factors in those first couple of years. You can really see the difference in the composition of the microbiome. In those first 3 years of life, as you phase from breastfeeding or milk feeding onto a whole food diet, there's kind of this expected phase that we go through where we have a really overwhelmingly high abundance of select bacteria like bifidobacteria. And then we kind of slowly start to phase into this much greater diversity and this rainforest-like diversity of the microbiome in those first couple of years as we start to phase on whole food diet and continue to develop. And so what we see is when you compare cesarean birth to a more natural birth, the timelines are just a little different. The microbiome position is a bit different. We don't necessarily know what impacts that has throughout the rest of lifetime, but it's really a question of, all the things that you couldn't control aside, how do you help the microbiome catch up to what we would want it to look like in a scenario? There are going to be mums who can't breastfeed, and that's normal. That's a lot of mothers will go through that. It kind of depends where you live and what kind of access you have available in those scenarios. Maybe formula feeding if that's the case, or type of formula. There's a lot of formulas coming out more recently that have what's called synthetic human milk oligosaccharides. These are these oligosaccharide molecules. They're like dietary fibers, but the HMOs, dietary fiber that's produced naturally by mum in breast milk. And those are really important during those first couple of years of life. Big studies like the Child Consortium have shown how important these are. And these plant-derived fibers like fructooligosaccharides and galacto-oligosaccharides, the FOS and GOS, they're not really the same. Yes, they're kind of a prebiotic molecule. They're intended for the same use, but they're really not the same, especially in daily life. So if you can find an infant formula that has these HMO molecules available, synthetic is fine, fantastic. These formulas still don't have all the different components, though, that you would normally see in breast milk. So there are certain places where breast milk registries are available, where other mums are able to donate any extra breast milk for those who are kind of struggling. I think that's kind of a perfect scenario, is that you can at least get a whole breast milk as an option. But if you can't, that's okay. Work with what you've got. And so I think, consider these antibiotic exposures and other things in early life. How do you expose your baby to as many microbes in early life as possible? You think back to, I'm sure growing up when you were a little one as well, your parents will say the same. It was quite commonplace that when a baby was born, you'd have a big family get together to introduce baby to the family and friends, and baby would get passed around to every auntie, uncle, grandma, friend, you name it. And it's important to remember that each of these people has their own unique skin microbiome. And so they're kind of exposing baby to all of these different diverse types of microbes. So that's one option, and allowing them to kind of play in the dirt and get exposed to these different environmental microbes. Being around pets, that's really been shown, exposure to having a pet dog and other pets like that has been shown to decrease risk of developing asthma and IBD and some of these chronic immune diseases. So again, kind of just allowing them to have that little bit dirty exposure to their environment is really allowing them to be exposed to these different microbes within their environment, helping them kind of establish that in their own body. So it's not a perfect world. We really want to be born into the mess. It's really normal for a baby during a natural vaginal birth to be exposed to different microbes from all of the different fluids, including stool, mum's poop, that they're going to kind of see this wealth of microbes. And from there, just not letting it stop, letting them kind of be exposed to a bunch of different human being microbiomes from all these different friends and family members. From there, you kind of start phasing on to whole foods, right? So now you're kind of, diversity is key. There's all kinds of different plants and fermented foods that kind of have this wealth of, again, microbes that are all in the food, but also, this breadth of fermentable carbohydrates. So those are starches and dietary fibers. And that's the food source for these different microbes. And they're quite picky little creatures. Each of them has a different type of carbohydrate or a few different types of carbohydrates that they prefer most. And so making sure that you've got kind of this diversity is the key to life. Well, it's the same for the microbiome. Each of these microbes prefers a different carbohydrate, a different fiber, a different starch that they like to eat that allows them to thrive and survive. And so providing a real diverse amount of different food exposures within early life is really essential to allow the promotion of these different microbes to continue growing and establishing themselves in the gut. So, Dr. Megan Rossi, she's a dietitian professor in the UK, big name in this field, really great at kind of discussing food, gut health, and the microbiome. It's this kind of core of 5 fiber-containing foods. So you've got your grains, your veggies, fruits, your lentils and beans, and your herbs and spices. And they all kind of contain types of dietary fibers and starches. They all contain all of these other beneficial phenols and proteins and other nutrients, and really trying to get as much diversity into your child as possible in early life, promote the growth of all these different microbes and kind of generally healthy nutrition, a bunch of different nutrients getting into the infant at the same time. That's kind of one of the easiest steps to promoting this expansion, this diversity of the microbiome. And so that's kind of somewhere that we focus in on our team too, is trying to understand, what is the best nutrition to get in early life, especially if you've got a mum who has given birth to a child, but mum had maybe a chronic autoimmune disease, or if she's on prescription medications or something that really can't be stopped during the pregnancy. And so you do already have this opportunity for the microbiome to already be quite limited if mum is the only person the infant is exposed to. So how do you promote that expansion of the microbiome and this training of the immune system and development of the organ systems in the baby through other environmental exposures to make sure that mum gets as much help as she possibly can with that transfer from mum to infant? That's kind of what we focus it on.
Right. So you have, multiple roles, multiple hats that you play. So one as, an academic at University of Alberta, and also the scientific advisor to Koenig. So how do you split your time? And I guess my question here is, what research are you focusing on in Alberta, and what projects, what products are you helping to build at Day Off? And maybe before we jump into that, just wanted to pull on that thread of the kind of year before the baby's born, right? So the parents' health, the epigenetics in a sense, what they're bringing to the table in terms of their own microbiomes and thereby the exposure. And just kind of wanted to echo that sentiment of, hey, it isn't a linear progression. You remember the work that we did with Rob Knight previously, where he created that beautiful principal component analysis of the case study that we did of how the gut's colonized. Is like, first, it looks like more of a vaginal microbiome. That's the baby's gut we're talking about. And then the skin from, like you say, passing around. And then eventually to this so-called, healthy gut microbiome, which is full of richness and diversity and a particular kind of evenness and array of, dare I use the word, species of bacteria that becomes the fingerprint to each individual. And so, in as much as it is unique to the individual, there's certainly, some first principles, and among them, obviously, the diversity. But you start to kind of see, particularly when you get into the issue of, different cases or clinical conditions, it's like, here's what an unhealthy microbiome looks like, for example, for people with, say, Parkinson's disease, or people with IBS or IBD, or obesity, type 2 diabetes. And so then what is this? Is this kind of a chicken or the egg type of thing? Is it, kind of a health issue that then the comorbidity is reinforced because of the reaction to the microbe? Or is there some kind of pathogenic infection, like, say, for example, like C. diff, that just obliterates the genome? And it even doesn't have to be so catastrophic. It could be like food poisoning, right? So you think, for example, I think we might have talked about this before. I always had this inkling that I don't think that the appendix is a vestige. I was like, I don't think that's doing something. I think that's doing something. I think, as we both know, it's been, it's a repository of probiotics, right? And so you can actually see, like, okay, there are some cases here: C-section, antibiotic use by necessity, also, appendicitis. These are issues where there's going to be a major perturbation. Perturbation is not even the right word. Perturbation before was like introducing solid foods. Now this is like, this is an obliteration of your microbiome. So I would suspect there's probably some kind of something that we can be doing interventionally now. Like I think, for example, we already know there's like vaginal swabs for babies who are delivered by C-section, for example, so that they can get that exposure. And you talk about these different food sources like breast milk donor banks. But, in terms of what you're seeing in kind of the clinical setting, and then even the human performance setting on the flip side, because I know you do some work with athletes. So it's, would love to hear how the work you're doing in the lab is actually translating to the real messy world on the business side of things.
Yeah, for sure. So, I think any of us who are in both academia and trying to balance business will say it's tough to balance, because academia is 100% our day job, and it really is quite demanding at times, for sure. But that they're 9 to 5. So it gives you some freedom outside of that to certainly be involved in, other business avenues that are of interest, I guess. So from a research perspective, you touched on it. It's all just so fascinating. And I think our group is really, if you were to kind of label us as what we're best at, we're kind of physiologists. So we like kind of understanding a bit of everything and putting it all back together into a system through systems biology. And so because of that, I think we are really interested in, not just one disease. How does this impact, one disease, one organ system, one cell type? But we really want to understand, how do you kind of put it all together, and what does it mean in both health and disease, and how all the different organ systems work together. So, you've nailed it. We all recognize that the appendix is, supposed to be a storehouse for probiotic microbes, these healthy, beneficial microbes that when you kind of ravage and disintegrate the microbiome of the gut, and then the infection passes, or the stress passes, or the life event passes, whatever it may be, the appendix is supposed to help reestablish the healthy microbiome of the gut. But there are kind of these disease settings. Diabetes is a great example of it, where they've shown, you can take the appendix out and it's actually protective against developing these diseases. And so there's kind of this question of, during the development of this microbiome, store health in the appendix in early life, is there maybe something going wrong in these disease settings where, it's no longer a probiotic backdrop. Now it's, pathobiont. And so it's not really pathogens. These are microbes that, they're probably fine, they're probably safe on their own, but if given the opportunity and the right environment, they can become pathogenic and they can kind of do some evil. And so, having a high pathobiont load in the appendix in these certain individuals seems to kind of predispose them to developing certain illnesses and never really being able to get over this unhealthy dysbiotic microbiome, because it just keeps being reestablished over and over again. So the appendix certainly has such an interesting role in all of this. And exactly that, what is it about the development of the microbiome that allows us to avoid illness and generate, the optimal human being? Athletic prowess is certainly such an interesting part of this. I played a lot of competitive sports through varsity and other organizations when I was kind of coming through school. And so the Dayhoff group as well, I think we all kind of have ties to pretty high level athletics. And so we're all quite interested in, exactly that. Like, is there kind of this role for the microbiome assisting with injury prevention and injury recovery and just that general athletic prowess and kind of overall best quality function of the human body? And the same can be said for even intelligence and other prowesses in life. We recognize the role of the microbiome in development of the central nervous system. You mentioned Parkinson's, certainly kind of the central nervous system and brain diseases that we know that the microbiome plays a role in. So the same can be said, if you take it the other direction in health, what is the role of the microbiome in, the most optimal functioning of the nervous system, including human intelligence and human functioning, high capacity functioning, quick thinking. And I mean, this all kind of comes back to athletics then. How agile are you? How quick can you make a decision? How quick can you see where the play is going and completely change what you were planning on doing? And so, that's from a corporate perspective, we've really been quite interested in the early life microbiome and understanding that is the best time to be intervening to establish the microbiome. And certainly there are other ways of changing aspects of our life, health and sleeping, physical activity, mental health and meditation, nutrition, to try to rebalance the microbiome. But we know that past kind of those early years, that kind of 3 to 5 year old range, you've kind of got this fingerprint set up already. So is the best time to intervene to create the optimal functioning human being really in those first couple of years of life. And so from a corporate perspective and the work that I've been doing, Dayhoff as the scientific advisor is, that's the best time to intervene. How do we define what is optimal microbiome, not just composition, so who's there and how much, Did you see a lot of bifidobacteria? That's always been a big question. We're running to the superstore, the best yogurt that has the most microbes in it. And that it's so much more than that, this diversity of microbes, and not just who's there, but what is their functionality? Are they surviving and really highly functioning in the gut and doing the job that they're supposed to be doing? So, we already touched on this. And I think we recognize that, you can have all the right microbes in the right amounts, real amazing diversity of microbes present in the gut. But if that environment that they're living in is not optimal, then they're not going to be as highly functional as they can be. And they're no different than human beings. You take a community of human beings and you put them in this beautiful, sunny location where everybody's happy and thriving and really loving where they live. And then you take them and you put them somewhere where it's always rainy and there's no sunshine and there's nothing to do, even in a workplace, they're not going to be the most highly functioning human beings. Well, the microbiomes are different. So if you've got an environment in the gut that's not perfect, it's not perfect pH, it's not the perfect nutrient balance, not the right metabolites communicating with the microbes from the host, then you can have the exact microbes that need to be there present, but their functions may not be as optimal as they could be. And so that's kind of a big approach that we've taken from an industry perspective is, lots of these tests that can be run on the market right now really just do very basic profiling of which microbes are there and how many are there. And then they'll kind of try to define, well, to fix that, take our supplement or our product. And we've taken quite a different approach because we're primarily bioinformaticians and scientists as a part of this group. And we've really taken the approach of, can we identify what your fingerprint is, not just, what does the composition look like? What does this community look like? But how functional is it? Is it responding the way we expect it to respond? Is it producing the right metabolites? Is it happy and healthy? And if it's not, if there's kind of subtle things that are perhaps not at its peak performance, what can we do to kind of promote change within a natural lifestyle? So whole food eating, what types of foods could we be promoting? Could you be meeting with a psychologist or a dietitian to kind of help you get back on track? And so, we really haven't tried to sell a product to be your one quick fix. We've really tried to take the approach of exactly that, and the wealth of really extremely incredible data that's coming out of some of these studies. Some of the individuals you mentioned, the likes of Justin Sonnenberg and some of these others who have really done these incredible studies that just show, the breadth of variety of plant foods that you're getting in really seems to support this diversity of the gut microbiome. And take that away, you really lose that diversity and that functionality. So can you promote whole foods, getting a good sleep that's successful for you, and real lifestyle changes? So I think they really marry hand in hand, the things that I'm quite interested in from a research perspective, and I'm trying to work with my team through the university to really understand what's happening and why and how to fix it. That kind of lends hand in hand to this industry side of things where it's a question of, if you're going to test it, if you're going to try to identify what's wrong, how do you do that without creating a scare tactic of, you've got one microbe that's not as high abundance as we expect it to be. Well, don't be afraid of one microbe being out of whack, because it's such a massive community of thousands and thousands of different microbes, and they have so much overlap in what they all do that it's okay if one or two are wrong, as long as their friends are still there doing what they're meant to be doing and kind of making up for that slack. So that's kind of the approach that we've taken both in our research and in our industry work.
And what are you seeing in terms of, let's call it customer response, to the extent that you can share? Because I know you're seeing, actually first, let me back up in terms of the first 3 years of development. Interestingly, my wife and I have been reading through this Nurture Revolution with Dr. Greer Kirschenbaum, and it's different but related. We're talking about like the emotional brain and the development therein, and kind of the first 3 years of this kind of neural plasticity and, well, if you will, training of the emotional intelligence. It's, perhaps it's coincidental that this is a critical time for the foundation of your microbiome, but I wouldn't say it's coincidental, correlational. I can't see there must be some kind of causation or relationship also. So insofar as, parents who are looking to you to help them, I suppose they're probably coming to you because maybe they know their gut, their children, they're are out of whack for the reasons that we talked about before, maybe medication, maybe C-section births, et cetera. I'd be interested to know how those conversations are going, what kind of results families are getting. And then on the extreme side, and again, I know a lot of your clients are confidential, what are the athletes looking for? What are the professionals looking for? Well, and what are they getting? Like, really, we preempted this call saying, well, hey, we're really at the tip of the spear. We're really in the infancy of the microbiome. But, clearly people are looking for information about their gut microbiome. They're taking action based on the insights they're getting. And so why or how is that information, so valuable insofar as we haven't been able to look through that lens before? And so what kind of recommendations are they getting that are different, and are they better? How is that, or is it all still unfolding? So how does it look like, I suppose, like with boots on the ground?
Yeah, I think that's a big part of what we all need to understand, from an industry perspective and even a medical perspective. When you see people coming into a clinic and seeking help from medical professionals, people want answers. And I think it's really hard to say to people, there's still a lot that we need to try to understand through research. So, we can do the best of our abilities for what we know today based on the work that's been done by many incredible researchers around the world. But it is, the microbiome field is really still quite in its infancy. I don't think anybody has really been able to perfectly nail down what is the optimal microbiome. And I think that's because it's such a symbiotic relationship between us and our microbes. And so it's so individual that it's really hard to take a group of people and say, what's the same about all of these people, when we know that there's such kind of personalized interactions that happen. But that said, I think from a disease perspective, and particularly when you're dealing with, families coming in who know that there has been a significant exposure or know that they have a risk based on mum or dad's health already, I think that's quite a bit different, because we are really starting to understand more and more each year these big changes that happen in the microbiome and how they are involved and linked to disease development. So the GEM Consortium is one really neat study that's been running out of Canada. It's under Ken Cortore's team in Toronto. Lots of teams around the world involved in this study. They were looking at first-degree relatives and the risk of development of Crohn's disease, which is an inflammatory bowel disease. And what they've been able to show is well in advance of development of this disease and diagnosis of this disease, there are these changes that are happening in the microbiome. Now, you've already touched on this. How much of a chicken and egg scenario is this? Is there something about the human being that changes first and that really promotes these changes that happen in the microbiome? And does it just turn into this perfect storm event that then culminates in the development of disease? That's really still quite hard to say, and it's really hard to study because, how many thousands of people do you have to profile before you manage to capture someone developing an illness when we don't know what causes it? So how do we identify who's the best person to recruit to these studies? So they're quite hard to run, but there are a couple of teams in the UK at the moment that are starting up a study that's quite similar. And there are lots of studies like this around the world. Child cohort is another great examples where, we're taking babies from birth and following them and just really asking the question of what allows a child to survive and thrive but not develop any health concerns, and what is it that results in allergy, that results in different disease issues, that results in different neurodevelopmental problems, learning behavioral problems, emotional stability, all of these different questions that we can ask. What is it about the microbiome and the human that culminate in what we would consider perfect health? Now, when you look at the microbiome and really kind of start to ask yourself, what is it about this that we actually understand right now, and how do we use it as a clinical tool? How do we actually answer these questions when families kind of come into all of these companies? We are not the only one by any means. There are lots of companies that offer gut microbiome profiling for adults, for infants, you name it. I think something that's really important to understand now when you're considering the product that you're purchasing is, firstly, how deep of a profile are they providing? So like I mentioned, are they only looking at the very basic level composition of the microbiome and they don't really care about how it's functioning? Or are they giving you a really deep array of understanding all the different microbes that are there, the different clusters that are important, and being able to say, it's okay if you're missing one or two, that's just your natural microbiome, that there are other microbes that have made up for that. And that's okay too. So as long as they're functioning in conjunction with each other, we're still good. We're still on the right track. So that's really important to consider. And I think that's something that I've really promoted the day off group to take into consideration when developing their testing kit that they offer is, seeing one microbe change or a reduction in the abundance of one microbe isn't the end of the world. So don't scare people into thinking that they're going to develop disease or they should be really frightened of their child's health if they see, one or two microbes changing. It's really a whole community event that you need to consider. The other aspect of these different testing kits that are available at the moment is if the kit is only being taken at one time point, how much does that really show you what this fingerprint is? Because we all know, any of us who work in this field, we know, if you were to profile my microbiome when I wake up and then again at lunch and then again at night and then again tomorrow, it looks completely different. And that's really going to come down to how did I sleep? What did I eat? What did I drink? What did I do? Was I sitting? Was I running? Was I stressed? Was I not? There's so many different factors throughout our day that impact what that microbiome community looks like and how it's functioning, because it's trying to work with us. It's trying to mirror us. It's trying to go through the flow with us each day. And so if these companies are only profiling the microbiome at one single time point with one single sample, what is that really going to tell you about yourself and your microbiome? And can that really get you answers? Or, the day you took that test, did you open the kit and now you're stressed reading through the instructions and you're worried you're going to mess up taking that sample. And so now you're already starting to impact your microbiome just in the sampling process. So you really want the company that's taking multiple tests when they run these kits and quite close together for the first sampling kit. So that's kind of something that the group, again, there's something that I kind of promoted the group to consider when they were setting up these tests initially. But I think at the end of the day, when we ask, why do people want these tests run? What are they actually being used for? I think there's so many different reasons, some people can be asked, you've already started teaching your kids about how to study for the MCAT and get into medicine by the time they're 10 years old, because that's your goal. You want absolute prowess in your children from day one, whereas other families, they really just want a happy, healthy child and they just want to make sure that they're going to be okay. Other parents are aware that they may have a health concern themselves that they're dealing with and they just don't want to risk passing that on to their child and they want to give their child a fighting chance. That, they really wanted kids and they're happy and thankful they could have them. So I think everybody kind of comes in with a different reason behind wanting to understand their own health. I mean, 23andMe was a great example. Everyone really wanting to understand where they came from and what their health risks are. And so I think we all kind of have our different focuses and our different goals. And the microbiome is no different. People really want to understand, what can we do to the best of our knowledge today to promote the most diverse and healthy microbiome possible. And so I mean, that's been the take home for people that we've worked with through clinical trials and science and things like that in the past is they come in for various reasons. When we recruit to clinical trials, it may be a disease study that we're trying to understand, or the interventions that we are promoting or providing around the microbiome is just how do you promote returning that microbiome back to as healthy and stable as we can for that individual. But a big part of that, again, is, the field has a long way to go still yet to understand what is perfect health of the microbiome and that interaction that happens between the microbes and the human being and how to comprehend and profile the personalization around that and the individuality, really respecting the individuality and that, you're not looking to make everyone's microbiome look the same because that's never going to happen. So I think, those are kind of some of the really big take homes.
Yeah. Well, it is one of those problems, right? Where, and I don't think it's a problem is not the right word, but you think about all the applications, right? So we talked about expecting parents. We talked about, chronic illness and inflammatory conditions. We've talked about mental health challenges. Aging population is another thing to consider. We talked about the athletes or, performance optimizers, people who have had exposure to chronic medication like antibiotics. Then there's just people who are biohackers and interested in this. And, we didn't even talk about children in developmental or behavioral conditions. And then, there's obviously the proactive preventative. And then you've got the whole slew of kind of healthcare, functional med docs kind of coming on board here as well. And so it makes it hard to, and then you talked about the importance of the temporal analysis. It can't be a one and done. Like the work that I'd done previously on the infant case study, is like we were sampling every day, multiple points of analysis. But okay, it seems like it is moving to a place where, okay, I'll tell you what I struggle with is it's not so much that the data is, it's not so much that the data is inconclusive. That's it's not the right sense. What I'm trying to get to is, it's not until you take a step back and you see the trend, right? Like, so let's use the analogy of your resting heart rate from your Fitbit or whatever Oura favorite wearable, or your HRV is a better example, because that's a heart rate variability, because that's much more of a finicky measure. And for the longest time, it was like, can I even trust this measure? And, there are a lot of these like high performance organizations that, investing into this, because they thought, well, what we want to do is we want to understand this HRV thing more, because what we know is when it's lower, there's a higher risk of injury. And when it's higher, that seems to be where performance happens. And so now we know we can't have a high HRV all the time, because that's just impossible. But could we align it up with the day of the Olympic final, right? Could we use this signal beneath the surface, you proactive, preventatively, predictively prescribe rest, right? Because sometimes we don't say how we feel, or sometimes we suppress our feelings. And anyway, so where I look at that is with athletes, it's like, okay, well, you're saying you look good, and you do look good, but then there's this metric, assuming that we can trust it, what's going on, right? And maybe it's nothing, maybe it's an anomaly, but what I've just said is it's not. This is a real data point, but the point is we're having this conversation. Right? I had a great epic coach, track and field coach, who I worked with him for 3 months. I was a walk-on track athlete, so I was raw, but I went from being ranked 700th in the country, this is varsity, in the 60 meters, highly competitive event, everybody does it, to working with him for 3 months and being ranked 7th in the country. Well, okay, so there's something that he was able to do in terms of unlocking some kind of latent, natural, innate capability. Because how much training do you really give 3 months to bring about such? None. I mean, even steroids wouldn't get you there. But he, well, what was he? He was a, when I started working with him, he was a 70-year-old Trinidadian sprint guru who was also like a, PhD in geology and psychology and human performance. He's more than a polymath. He was just a lifelong learner. But what he was really good at doing was seeing people, asking questions, and getting to the root cause that a lot of the times was, stopping, in this case, athletes from performing. It wasn't an athletic issue, as it turned out. So I guess my point is, like, something like HRV and a good conversationalist, somebody who knows how to, not necessarily use this as a diagnostic, although there, certainly are things that we could use gut microbiome tests for that would be diagnostic, like Clostridium difficile, for example, to have let's say the temporal analysis as something that we can look back to in hindsight. Like, I see the true opportunities, like, well, it's the metagenomic problem. This is hypothesis outcome. When we look back, we can see here all these correlations, and then we can do further tests to discern whether or not is causation. But nevertheless, the blocker to that, like whether it's you test once a year, once a quarter, once a month, or every day, seems to me that insofar as, like, say, optimizers, let's say, or even parents who are dealing with, a child who's failing to thrive, that's a lot of work to do all the sampling, right? So, like, is there a future where, just kind of go out on a limb with me here, because I want to kind of two questions. Like, one, is there a future where it's more automated in terms of the collection? Like just put it in the toilet, You know what I mean? And you get the family microbiome every day. And if it's like, if something comes up, well, we got to break it out and do a follow-up diagnostic. I like the idea of this like house microbiome. So you've got like a filter is running through. Let's run the metagenome of your house, your microbiome of your house. So like, again, to me, it seems like the limitation isn't in the signal necessarily. It's kind of in the collection process, right? So
Technologies.
yeah. So do you see any companies making strides there? Like, just make this easy. Like, I mean, I designed I orchestrated the collection process from the 500 twin cares and what we were working with there, you wouldn't believe. It was this insert that you put on the toilet seat, and what a nightmare. So, I mean, we've come a long way. Like, I look at all these commercialized versions of this, but it's still too much. It's still too much. Just like going to a physio is too much, and they see you out of context, right? It's like, this is not how you move in the real world. So it's like, I mean, I envision something just like that. There's like a natural kind of collection in your toilet, and sent off to the lab once a week, maybe once a month. Are there any companies doing that? I mean, this can't be the first time somebody's thought of this.
Yeah, I mean, wouldn't it be incredible? I think the big issue right now, and I won't knock any of them, because the incredible strides they've made to make sequencing cheaper, faster, more accessible. It's just mind-boggling what's happened in the last decade or two.
Yep.
And you see some of these companies that are striving now towards, particularly in veterinary medicine, actually, striving towards kind of on-the-go sequencing in the back of a van, so that you can go around to some of these like large industrial farm sites and profile animals and their microbiome and things like this. So, I mean, the technology, I think the advancement and the pressure is there to find more easily available sequencing technologies, advance the speed while maintaining the accuracy, and kind of allowing for some of these home sampling kits. I think at the same time, you see other industry groups, and we are among them, trying to improve just how easy it is to take a sample and submit a sample while we wait for the sequencing speeds to improve. Because right now, realistically, the problem is the proper, accurate, deep sequencing that provides both the composition and the functionality of the microbiome. It takes probably 2 weeks to process the sample, sequence the samples, get all the data, and put it back together again. And so how do we start automating that process? But in the meantime, are there ways that make it much easier for someone to take a sample and ship it in that really doesn't take much time at all? But I think you're right. I think that's the dream that we all have is, at what point does this turn into, I tune to diabetics and the fact that they can prick a finger, put the test kit on it, and boom, what your blood glucose is, and you've got a machine that's literally pumping the exact amount of product into you to make sure that you're completely balanced at all times. That would be the absolute end all be all, is just to be able to take these daily samples and have it say, this is what's for breakfast today, because this is what your microbiome looks like, and this is the precision that we need to maintain your health.
I think there's the passive data collection that I think of, that like, how do we make it more convenient for people? Like, okay, I've got a diaper bin for my daughter, so it's like, hey, here's all you got to do is write the date and the time on the nappy, and put it in its bag, and then just send us this bag once a month. We'll do the rest, right?
Yeah,
that would be a shitty project. But that's kind of the joke that I use with the Cornell is like, it's messy, but somebody's got to do it and willing to do it. And, I think.
And that's where we're at, right? So, I mean, like doing listing kits, that is exactly what we're doing, is you take 3 samples within a week of each other, that kind of your general life print of the week. And, once every couple of months you repeat to see what it's looking like. Are you seeing?
See, I think that's, yeah. And that's, I think if you're making it clear what the outcome is, it's like, okay, you're going to do this, and here's the before, here's the after. So on the flip side, it's like, let's use a compelling case, like, well, like astronauts, for example, right? Like they're already monitored anyway. So where are those? I mean, I would assume there's research going on in those special groups, and how do we use those insights? How do those come to the frontier? How do those, general insights deliver us, I don't know, say more informed, updated principles on how the microbiome goes? Like we've already done this with, RDAs and, soldiers who went off to war and suffered from malnutrition.
Yeah.
Yeah. Yeah. And it's a really careful line to walk, because you can't be unethically experimenting on folks. But I guess what I'm saying is where is the passive data collection already happening, and how do we leverage that?
Yeah.
Yeah. Okay.
Yeah. And I mean, athletes are no different, right? So I mean, athletes get tracked so closely, and that kind of
what
we've been working with is athletes get tracked so closely for, what happens when you retain an injury, or what happens during your post-injury recovery phase, and, what was it about you that made you come back quickly, that kind of delayed your recovery phase, and like the likes of work that we're doing with some of our retirees, our professional athlete retiree groups, is they really want to understand, particularly in some of these high contact sports, what was it that put me at risk of developing a brain injury that wasn't just a concussion, but turned into now like quite an early onset dementia? And was there something that I could have done during my athletic career that would have put me at lesser risk, or faster recovery, or more successful recovery, and kind of reducing that risk of outcomes beyond their athletic career that now follow them for the rest of their lives? Because that's one of the big concerns that we see with our athletes that we work with is, as you're kind of following me throughout my absolute peak performance, when I'm having the best games of my life, the best seasons of my life, and like I said, I mean, the microbes have been associated with your ability to think quickly, make decisions, avoid these injury scenarios. And then so much as the recovery phase, how quickly do, these skeletal injuries heal? What about the musculoskeletal general injuries? What about these concussion injuries? And the microbiome's really been linked to recovery in all of these phases and outcomes, chances of kind of returning back to your complete original prowess versus having this injury that now plagues you for the remainder of your foreseeable contracts that you have left in your career. And so, even these like incredibly high functioning individuals, what is it about both the human aspect and the microbiome aspect that allows them to continue doing what they're doing, or kind of reduces this risk of, not just developing a disease, but from injury and maybe.
Yeah, and it is something that does, once you've got that information, it does lend itself to a therapeutic, right? So in the case of, the astronaut leaves Earth with a certain microbiome, that diversity is going to be depleted, you would think. So it's like, hey, we've got your probiotic blend coming back, because we know that gut transplant therapies are effective for, I read that that's true for things like Parkinson's. We've seen, striking phenotypic changes in mice with respect to obesity, lean, and then also mental health as defined by anxiousness in rats. So the idea of like being trained up as an athlete to the peak of your performance, and then, okay, I'm going to collect a bit of my microbiome here. And it was like in Rio, right? There was concerns about the water quality or yeah. So it's like, okay, I'm going to travel with my probiotic. Well, heck, I mean, if you're an astronaut, if you're on Elon Musk's space mission to Mars, I probably, want to have a supply of your healthy gut microbiome on board, because you're not going to be inoculating on Earth, that's for sure. So, gosh, we've taken a number of twists and turns here, and I want to be respectful of your time, because I know you're just coming off on a whirlwind of scientific, let's say, exploration with conferences, and I know you've moved, and I know you've got big things coming. So wanted to circle us back, Heather, just quickly on, my favorite 3 questions is, what's the key message here for our listeners, and who needs to hear it most among them? And then of course, number 3 is who would you recommend as our next guest on the DNA of Things?
Yeah. So, I mean, the key take home is just that those microbes that live inside of us they're our best friends. They're going to help us not just thrive, survive, and gain nutrients, but they are the key to longevity, annual life, athletic prowess, intelligence, and overall health. So, they are your best friends. Treat them wisely, feed them wisely, and make sure that your lifestyle supports their health, not just your own. If you start to lose them, it's hard to get them back. So really show them the respect that they deserve too. So love your microbiome. And I'd certainly say, I think this is applicable to pretty much anyone. I think we all have kind of our own health concerns, our lifelong concerns, the things that we are aspiring to complete in our lives, be it career or athletic or, having a family and making sure that they have the highest chances of health and success. So, any aspect of a human being, we all have our different goals. And I think that really comes back to not only our own health, but the health of the microbiome. So I think really anyone who's interested in peak performance and whatever that means to them, I think it'd be of interest to really understand these types of topics. And in saying that, it really depends on the type of topic you want to run to next. I think Justin Sonnenberg and his team have such neat studies going on the microbiome and how it can be impacted. Jens Walters over in Ireland has this new NIMI study that's coming out of Papua New Guinea and looking at kind of how can you impact the microbiome and feed it through nutrition, and how do we really get back to our connection with the earth, and really looking at kind of some of these remote communities and how they kind of live and look so differently to what we do in what would be called the Western world now. So I think those are kind of some of the guys that I follow most. But yeah, there's a lot of pretty cool people doing some pretty neat stuff. So.
Well, I'll definitely circle back with you for intros to those folks, and we can certainly extend the invitation. So, well, thank you, so much for that recommendation. And, Heather Armstrong, thank you for, sharing your insight. Not everybody is spending the time and doing the diligence that you are. And so, I'm really grateful that we could bring some of the great work that you're doing to light here. And I'm really excited to share this with the audience and to, pick up our conversations with the DayHop folks as well. And actually, just a final note is how do people find you if they want to connect with you or your company? I'd love to pass all that information.
Yeah, for sure. So it's a pretty quick Google search to find me academically. It's just Dr. Armstrong at the University of Alberta will pop up with my profile and my email. Pretty easy to get in touch that way. From a corporate direction, they have technologies, order the test kit from there. There's contact information if you're interested in, how these kits are used and what kind of potential there is there. Pretty easy to find them online too. But those are probably kind of the two easiest ways to get a hold of me.
Amazing. I'll put that info in the show notes. Dr. Armstrong, thank you so much for your time and joining me on the DNA of Things today.
Thank you for having me. It was really great to see you again.
You've heard the science, you've felt the story, and now you've seen the thread. It's not just in fecal samples or breast milk or gut tests. It's how we feed our children, how we recover from wounds, how we perform under pressure, and how we rest. The thread is real, follow it, and it may lead you to yourself or to the garden you forgot that you were growing.